this. Yeah, so it's just a continuation from previous lecture where we have discussed the counterpart of this one, which is prokaryotic DNA replication. So today we would be looking at some of the specifics of this particular process called eukaryotic DNA replication. Mind you, this process is very similar to prokaryotic DNA replication in terms of various components that it has, be leading strand, lagging strand, the involvement of a polymerase, involvement of primers, the presence of 3 stages: initiation, elongation, and termination. All of those are extremely similar. So, so we will discuss about that a bit more in detail. But there are, of course, differences, right? There are major differences between eukaryotic and prokaryotic polymerases, and we would be emphasizing a lot more on these differences as well in today's lecture. So after this particular lecture, we would be moving into some other territory that is completely different from replication. It's related to replication, but it's not a continuation of replication itself. But that's called DNA damage and repair. So at the end of this particular session, we would be moving directly into that one. But I'm not really sure I would do that today itself, because the 2 concepts might actually confuse you. So I might mostly finish eukaryotic replication and all the affiliated concepts to eukaryotic replication today itself, and then call it a day, and then pick up the thread and continue on from next week onwards when it comes to DNA damage and repair, because it's not exactly the same seamless transition from replication into DNA damage and repair. So you might get some sort of a confusion if I do that today itself. Right. So I really hope, before I move further on, I really hope that you had a chance to take a look at the Google Classroom, how that operates and everything. If you, I had at least one complaint about you not being able to join and not receiving the invitation or something like that. So what I did was that I resent the invitation. But if you haven't received the Google Classroom invitation, please check your spam box, because if it ended up in your spam box, it doesn't matter how many times I send it to you, it would go and still end up in your spam box. So it's best you dig it up from your spam box and try and join the class. So if you haven't received an invitation, please do that, and do take a look in your spam box, and it might be there. Right, and if you still have any difficulties in joining the Google Classroom, you should tell me immediately, right? Otherwise, it's going to be very problematic later on if you are not able to join the Google Classroom, because you will have the lecture recordings, all the lecture material, reference material like textbooks and things like that, and then educational videos and animations. You will find them there, and also I will also be giving you some assessments on Google Classroom. So be prepared for that. So you'll be using that a lot. Right. So I hope that is reasonably clear. So moving back into eukaryotic transcription, eukaryotic replication. One of the main components, or I would say the main component of any replication process is actually polymerase. So when it comes to eukaryotic replication, it's no different. The main thing, or the primary thing affiliated with replication is polymerase. So in this scenario, it's actually eukaryotic polymerases. So there are several different eukaryotic polymerases. So when it comes to prokaryotes, there are three main polymerases that were involved. We discussed that in one of the previous lectures. There was DNA polymerase 1, 2, and 3, and one of them we have designated as the primary polymerase. And in eukaryotic polymerases, when it comes to eukaryotic polymerases, it's no different. There are several different polymerases involved in the eukaryotic transcription. Excuse me. So starting from alpha, beta, gamma, delta, and epsilon. These are the main, there are 5 main eukaryotic polymerases involved there. But now, these are the main ones. Right? These are the main ones. There are 5. But now research suggests that there are even more. Right? There are even more eukaryotic polymerases. So at this point in time, in 2022, in 2022, there are at least 4 other eukaryotic polymerases are known, which are not really shown here, but it's those are rarely encountered polymerases. I mean, less ubiquitously encountered polymerases than these 5 listed over here. So these 5 are the main, right? But apart from that, up until this point over here, until 2022, 4 more eukaryotic polymerases are known, right? So altogether, there are about 9 eukaryotic polymerases are known up until this point, but there are 5 main ones, right? So these are, for the sake of convenience, these are named as alpha, beta, gamma, delta, and epsilon. So if you compare all these, they have different sizes. The size of the protein itself, they have different sizes, starting from alpha, starting from 250 kilodaltons, delta 170, epsilon 256, beta has a range 36 to 38, then gamma 106 to 200. And then the location of these polymerases, most of them are found in the nucleus. So the first 4 at least, alpha, delta, epsilon, and beta, they're all found in the nucleus. But then gamma nucleus is the replicating enzyme for mitochondria. Right? So although it's found in eukaryotic organisms, it's precisely found within mitochondria. Right? So if you actually look at this gamma polymerase, if you look at it structure-wise, if you look at the sequence that codes for this structure of this polymerase, if you take a look at that, and if you make a comparison of this polymerase to prokaryotic polymerases, if you make a comparison of this polymerase into prokaryotic polymerases, they have a very striking resemblance, right? They have a very striking similarity between the mitochondrial polymerase and prokaryotic polymerase. So this gives you a very strong hint, or some very strong evidence that mitochondria has been solitary prokaryotic organisms in our evolutionary history, and at some point during our evolutionary history, they have encroached inside eukaryotic cells, and they have taken up some sort of a vital role in energy metabolism. So it's very strong backing for that claim, because most of the enzymes and most of the proteins that you see in mitochondria, they resemble eukaryotic proteins and eukaryotic enzymes, and a lot of the sequences are also very similar. So that's a very interesting thing to look at. Some of them have 3' to 5' exonuclease activity. Some of them don't. So if it has 3' to 5' exonuclease ability, in other words, it can proofread and have some DNA repair function. Some of them have, some of them don't. So, for example, delta, epsilon, they have, and even the mitochondrial polymerase, it has the exonuclease ability. And alpha and epsilon, sorry, alpha and beta, they don't have any exonuclease. And the next ability is the primase ability. So, in other words, the ability of polymerase to put an RNA primer. Alpha has that ability. No other polymerase has that ability to put a primer, put an RNA primer. And I also list a few other properties of these polymerases: processivity, fidelity, so whether it's involved in replication or it's involved in DNA repair. So some of them are, for example, they are involved in DNA repair. These are involved in DNA repair. Some of them are not. Some of them we do not know. So if you do not know about that, there's a question mark, open question mark. That is open to debate. So when it comes to functions of polymerases, alpha polymerase, its primary function is to synthesize an RNA primer, and then which is also mentioned in this little table over here. It says primase activity, alpha has it, and no other polymerase has it. Polymerase delta, DNA polymerase delta, is the main polymerase in eukaryotes. Right? So this is the main replicating enzyme, or the main replicating polymerase when it comes to eukaryotic organisms. It's DNA polymerase delta. So when it comes to prokaryotic organisms, it was DNA polymerase 3, like that one. In the case of eukaryotes, it's DNA polymerase delta. And then there's another protein that acts as the sliding clamp. So we remember how the prokaryotic sliding clamp looked like. It looks a little bit like that. It wraps around DNA and holds the whole assembly in place, including the DNA polymerase and the replicated DNA strand. So it holds everything in place. So that's the DNA clamp in eukaryotic DNA polymerase delta, right? So this one has a special name. We call it pCNA. Sorry, we call it pCNA. So pCNA stands for proliferating cell nuclear antigen, right? Proliferating cell nuclear antigen is called pCNA. So DNA polymerase epsilon is another main polymerase that find in eukaryotic cellular environments. So this may replace polymerase delta during Lagrange synthesis. Okay? So it can interchangeably, it can sometimes change over. It can take over from DNA polymerase delta. Polymerase beta is another polymerase that you find very commonly in the eukaryotic environments, cellular environments, that's involved in the mismatch repair. Right? So it mainly has a repair function. Polymerase gamma is the last one. So this is the replicating enzyme of mitochondria, right? So this is the DNA replicating enzyme, or the polymerase enzyme of mitochondria. It has a very strong resemblance to prokaryotic polymerases. Has very strong resemblances to prokaryotic polymerases. So in other words, it's more closer to prokaryotic polymerases than to eukaryotic polymerases, right? So that's one of the main reasons why we think that these organisms, these mitochondria, they have a different evolutionary past compared to eukaryotic cells. And they have, encroached inside eukaryotic cellular environment at some stage in the evolutionary history. Now, one other main difference in differences in prokaryotic replication is that eukaryotic replication is that the prokaryotic DNA replication mainly involves a single replicon. So the origin of replication is just one. So it has a single origin or origin. Eukaryotic replication involves several replicons, right? Not just one, it involves many replicons. So it could be several, or it could be a dozen, or something like that, right? So the replication origin starts in multiple places. The other main difference between the two is that the speed at which bases are being added. So prokaryotic DNA polymerase adds 50 base pairs per minute, approximately. So that kind of approximation, on average, it does that. However, when it comes to eukaryotic polymerase, it adds only about 2,000 base pairs per minute. So it's a markedly slower rate. So it adds nucleotides at a markedly slower pace compared to prokaryotic DNA polymerases. Right. Now I think we would head right into eukaryotic DNA synthesis. So you know that just like prokaryotic DNA replication, it works in 3 stages, right? So just for the sake of simplification, I will discuss the initiation and elongation and everything with the aid of an animation. So I have simplified the eukaryotic DNA synthesis so much that could fit in a single diagram like that. So I will take you through some of the main proteins that are involved, just for the sake of simplicity, and with the aid of the animation, we'll also talk about some of the main a full breakdown. So a component-wise breakdown we will take a look at with the aid of a little animation. Because if I try to bring in all the little components over here, it will very easily confuse you. So I do not want that to happen. So I would want this to begin with something with relative simplicity. Right. So you can see that there are several different components over here. So once the initiation had begun, the replication unit, the main replication unit, or the main replication machine, the main replication machinery looks a little bit like this. So you have a helicase. So it has several subunits. We call them MCM 2 to 7. So the helicase is recruited over here. So we also spoke of DNA polymerase alpha, which can put an RNA primer, so that acts as a primase too. So that's polymerase alpha with the primase activity. And here you have the main polymerase, DNA polymerase delta, with the sliding clamp, which is called the pCNA, right? So pCNA is this bit over here, the one that looks like a little ring, right? So this is the lagging strand, right? So this is the lagging strand, and this is the leading strand over here. Okay. So Just like in prokaryotic DNA synthesis, in the lagging strand, DNA synthesis is discontinuous with the help of Okazaki fragments. In the leading strand, it's continuous without the help of any Okazaki fragment. So it's not fragmentized. It's just one continuous clean synthesis along the leading strand. But synthesis happens in a staggered fashion, staggered and a discontinuous fashion in the lagging strand. Now, the whole assembly is kept apart by special proteins called RPA. So these are similar to single strand binding proteins or SSBs in prokaryotes. So similar. This is similar to SSBs, single strand binding proteins in prokaryotic organisms. So this is the main these are the main components of the replication fork or the replication machinery. Now the leading strand synthesis is initiated by primase, DNA polymerase alpha, because it has a primase function, and then RFC mediates the polymerase switching. So RFC stands for replication factor C. Right? RFC stands for replication factor C. Replication factor C, what it does is that it mediates the transition from polymerase alpha to polymerase delta. Right? So once the primer is added, polymerase alpha adds a few more nucleotides. It adds a few more nucleotides, about 20 to 25 nucleotides it adds, and then it switches over. It gives its role to DNA polymerase delta, right? So it basically from DNA polymerase alpha, the DNA polymerase delta takes over after a short strand synthesis. Right? So polymerase alpha does the primase function, and it also adds a few nucleotides to the growing chain, and then it lets DNA polymerase delta take over. Now the whole thing is facilitated by RFC. Right? The whole transition is facilitated by RFC. Now, so that's clear. What am I saying here? Removal of DNA polymerase alpha, assembly of pCNA, and addition of polymerase delta is done by this one, right? And then DNA helicase unwinds the DNA at the replication fork like that, and during elongation. So when elongation happens, helicase keeps unwinding this way, and then once it keeps unwinding, we get the same problem of forming knots at this end, right? Forming knots over here. We have the same problem as prokaryotic DNA replication. So the remedy for that is basically a DNA gyrase or topoisomerase. So the DNA gyrase DNA gyrase would sit on top of this one over here. So a gyrase would sit on top of this one over here, and then it would untie all the knots that are forming in front of the advancing replication fork. So the replication fork moves this way. Because the helicase is continually unwinding, and the polymerase is continually synthesizing both in the end of the lagging strand and in the end of the leading strand as well. So that's happening. So both of these processes are happening. So the whole assembly is actually moving in this direction. So when the whole assembly is moving in that direction, DNA starts to supercoil, right? DNA starts to supercoil at this front end, and then it starts to knot, right? So to ease the supercoiling and to ease the knotting effect, you have a topoisomerase for DNA. So topoisomerase. Or another word is gyrase, right? DNA gyrase. DNA gyrase. Right. So I hope that is reasonably clear. So I'm mentioning that the DNA helicase unwinds the DNA at the replication fork, like so, like this one. And then on the lagging strand, DNA polymerase alpha initiates synthesis through formation of an RNA primer, just like that. This is the RNA primer. This little bit over here, that's the RNA primer. And then immediately following that one, it adds a few more nucleotides. It adds a few more nucleotides like that. And then with the help of the RFC, it switches from alpha, DNA polymerase alpha into DNA polymerase delta, right? So RNA primer is there. It's followed up by a short DNTP oligomer, which is this one over here. I'll use a different color maybe. So that's a little DNA oligomer. And then RFC loads pCNA and then second DNA polymerase, which is delta or epsilon. Sometimes it can even be epsilon, but 99% of the time it's 99% of the time it is delta, DNA polymerase delta. Right? And then the synthesis of the okazaki fragment continues. So the abbreviation is over here. RFC is replication factor C. Is a 5' subunit protein that is used as a clamp loader in eukaryotic replication. Right? So it recruits pCNA, and then eventually it recruits DNA polymerase delta as well. So it has a function of a clamp loader. So pCNA is the DNA clamp. So what loads the clamp is the clamp loader. So in eukaryotic DNA replication, the clamp loader is RFC. Right. When the DNA polymerase approaches the RNA primer of the downstream okazaki fragment. So we're talking about the lagging strand here. So it's the lagging strand. So in the lagging strand, when the DNA polymerase approaches DNA primer of the downstream okazaki fragment, RNase H1. So another enzyme comes in, RNase H1 comes in, and it degrades this short stretch of RNA by removing all but one of its ribonucleotides. Okay? So it removes everything, every ribonucleotide it removes, except for the last one, right? So the last one is being removed. So another enzyme is responsible for the removal of the last one. So that one is called FEN1 or RTH1, right? So quite a few enzymes are involved in this process. And then finally, the DNA ligase comes in and joins the two okazaki. Right, so I think it's fairly simplified and fairly easy to understand in this format. However, life is not as easy as we think, right? There's so much to this. There are so many tiny little other components that are involved in this whole process of DNA elongation, initiation, elongation, and replication. So let's discuss that with the help of a little with the help of a little animation. I must give you a little warning that it has so many different minor components. It can be a bit overwhelming initially. But I certainly do not, expect you to. The components that I discussed over here, these are the absolute necessities, right? These are the bare basic or the bare bone components of this whole replication process. So the components that you will encounter in the animation, all the other little bits and pieces could be less important to know because the main function is already described over here in these two simplified diagrams. But you will encounter so many more little other less significant components. It's broken up into so many other little components. So I do not expect you to memorize any of that. But what I invite you to do is to have a good understanding about this whole process, at least up until this point, or at least at this level, right? Because you cannot simplify this any further, right? So what I described in these two slides is the most simplistic scenario, right? The most simplistic. Okay. Right. So what I described over here is the most simplistic picture that anyone can give you in terms of eukaryotic replication. Right? But there are in the animation you will see that it's a bit more complex. So let's take a look at that one and discuss further.
Eukaryote. You know the replication process proceeds in three major steps: the initiation part, the elongation, and the termination. And here in this video, we'll have a detailed look on the initiation of DNA replication in eukaryotes. Now here you can see the DNA molecule. It's a linear one in eukaryotes, having two strands running in opposite direction as shown in the diagram. And also in this diagram, you can see a site called ORI site. Like we have ORI site in prokaryotes, in the same way we have ORI site in eukaryotes also. This is where from the replication starts, or you can say it is the origin of replication. This origin of replication is AT-rich region. Like we have ARS in yeast. The ARS is the ORI site in yeast. It's termed as autonomously replicating sequences. Before getting to the mechanism of replication, let's see what are the proteins or enzymes involved in the initiation of eukaryotic DNA replication. First, we have ORC protein. It is a complex protein known as origin replication complex. It binds to ORI site and has ATPase activity. This protein recruits all other proteins for initiation purpose. The second protein for initiation is CDC6 protein, cell division cycle 6 protein, required for helicase loading. Then we have CDT1 protein, CDC10 dependent transcript, also used for MCM loading or helicase loading. CDT1 interacts with MCM and induces topological association of MCM ring with the DNA. Then we have MCM2 to 7, that's mini chromosome maintenance. It's a multi-protein unit of 6 MCM proteins, which shows helicase activity. Then we have CDK and DDK, both are kinase proteins, cyclin-dependent kinases and DBF4-dependent kinase. Both regulate the initiation proteins by phosphorylation. Then we have CDC45 protein, cell division cycle 45 protein. It interacts with MCM7 and polymerase alpha. The CDC45 activates the helicase activity of MCM protein. Then there are genes, GINs. It is a co-activator of DNA initiation, forms CMD assembly, CDC45, mini chromosome maintenance, and genes, that is the CMD assembly. Furthermore, we have RPA protein, replication protein A. It prevents the enemy of DNA strands from separating. These are like SSB proteins of prokaryotes. Then we have RFC protein, replication factor C. It is a DNA clamp loader. It loads pCNA to the DNA strands. Then ultimately we have pCNA itself, a DNA clamp. It holds to DNA in a ring-like manner. And with this DNA polymerase does not get detached from the synthesis site. Now talking about polymerase enzymes. First we have DNA polymerase alpha. It synthesizes short DNA stretches on primers. Do not get confused here. This is not the main DNA replicating enzyme. It just initiates the first few deoxynucleotides on which then other primary polymerase will work. And this primary polymerase is the polymerase delta. It synthesizes both leading and lagging strands. And also we have primase enzyme which synthesize primers. Primers or the ribonucleotides. We need primers because DNA synthesis cannot start de novo synthesis. It cannot start from scratch. So a primase is needed which starts the RNA synthesis first in the form of primers. Then on primers, the primers are elongated with the DNA synthesis. So a primer, that is the RNA synthesis, can start de novo without the requirement of a primer. Then we have other enzymes for elongation like ligase and clap endonuclease. Now let's see the initiation process in eukaryotes in detail. Here in this diagram, you can see the DNA molecule with ORS site. The first protein it attaches is the ORC protein. The ORC binds to the ORI site. Then with the ATPase activity of ORC, it loads all other initiation proteins. The first two proteins recruited by ORC are the CD6 and CDT1 protein, as you can see in this diagram. Now the CD6 drives the loading of MCM protein, that's helicase enzyme. And the CDT1 induces the topological changes in DNA, with which MCM ring is associated firmly with DNA, thus concluding the helicase loading. So this forms as the pre-replicative complex. And these all events are going in the G1 phase of cell cycle. But till now, the MCM2 to 7 complex does not show any helicase activity. It's inactive yet. Now from here, there will be transition from G1 to S phase. And this transition is mediated by kinase enzymes like CDK and DDK enzyme. The CDK phosphorylates CDC6 and DDK phosphorylates MCM, that's helicase. And in this G1 to S transition state, the CDT1 is also inhibited by G1 protein. And finally, the ORC is also phosphorylated so that it does not recruit further initiation protein. Now the phosphorylated CDC6 protein is degraded and CDT1 is also inhibited. And in that process, the phosphorylation of MCM leads to the recruitment of CDC45 and genes proteins. And furthermore, we see the CDC45 is loaded to MCM with the help of SLD3, SLD7 complex. Now in all these processes, we have MCM protein on DNA bound with CDC45 and genes. We also have ORC here, but it's inactive, so we will not show this here. Furthermore, the assembly of MCM, CDC45 and genes makes us the CMG assembly. That is the CDC45 protein, mini chromosome maintenance, and GINs. This CMG moves across the DNA and melts the DNA in order to give room for polymerase enzymes and other enzymes. The formation of CMG assembly also recruits MCM10 and CTF4 protein to the DNA. And these two proteins in turn recruit RPA protein, polymerase alpha, and primase. And polymerase alpha and primase are always associated with each other. The RPA binds to both strands and prevents the enemy. If we zoom into the diagram, we can see MCM running towards the left and breaking the hydrogen bonds, thus showing the helicase activity. Then we have a primase enzyme, which adds primers, the short RNA stretch. Primase adds one primer to leading strand, while as it continuously adds primers to lagging strand. because on lagging strand, the DNA synthesis is always discontinuous. That's in focus fragments. Now, after these events, the polymerase alpha works on these primers and adds few deoxynucleotides to initiate the DNA synthesis. The polymerase delta needs few nucleotides first, which are given by polymerase alpha to kickstart the replication process. Now, from here, the elongation process starts. Like pCNA clamp attaches the strands and delta polymerase then associates with these pCNA molecules for elongation of DNA strand. So this is all about DNA insertion process in detail. In the next video,
Right. I hope it is reasonably clear. You must have seen that it's a fair bit more complex than I mean, there's a fair bit more complexity to it than what we have discussed over here. So what I have presented is the bare basics. So I would expect you to have an understanding up until what I have at least discussed to you with these couple of slides, right? In that little animation, it gives you the current understanding that we have, or the current working model of eukaryotic DNA replication that we have at the present. So, for an example, if you go into research after your degree into higher education, and if you end up in a research field that has anything to do with DNA replication and things like that, you will need to have that understanding, that full understanding that I have shown you, because that is the current working model of DNA replication that we have at this point in time, right? So these couple of slides have given you a simplified idea, but that animation had given you a full overview, right, or the current understanding of the working model of DNA with all the component proteins. So it described everything. So all the component proteins, including genes, CDKs, all the other component proteins, they were all described in that one. But for you to describe eukaryotic DNA replication in a mechanistic working manner, you don't need that level of detail. You can still describe it up to this level, but without this level of understanding, it's almost impossible to describe that. So that's why I said you should have at least this level of understanding when it comes to eukaryotic replication. Right, so now you have seen how initiation and elongation takes place. But what about how it ends? So we know that eukaryotes have linear chromosomes. They have linear chromosomes like that. And when they replicate, so let's see. So when they replicate, it forms a little structure like that. And it would end at one end, because it's not a circular chromosome like it's not a circular chromosome as in a prokaryotic organism. So there's no circular argumentation here, right? So it has to end at one end over here when it comes to eukaryotes. Now, this actually, so that's where the termination happens. But there's a very big problem. So I'll explain. So once I explain that, you will realize what it is. So there, it runs into a problem called end problem, or we call it the telomere problem when it comes to eukaryotic replication. So if you look at this particular image over here, so this is a fluorescence micrograph, right? It's a fluorescence microscopic picture. So this is the fluorescence micrograph of eukaryotic chromosomes, right? Eukaryotic chromosomes, and you can see that the ends. So these are the chromosome ends, right? So these are our chromosomes. So you can see that the chromosome ends are lighting up, right? They're actually glowing. All the chromosome ends are glowing. So the reason for this glow is that at the end of these eukaryotic chromosomes, we have special sequences called telomeres. There are special sequences called telomeres. So in this experiment, what they have done is that they have designed oligonucleotide probes, or that are complementary probes that are complementary to these telomere regions, and they have put a little fluorophore, right? So they have so a probe is like that. So probe is a complementary sequence, right? It's a little single-stranded complementary sequence like that. But at the end of the sequence, you have a little fluorophore. So a fluorophore is a fluorescent molecule, right? So if you shine a specific wavelength light on a fluorophore, so let's say, just for the sake of the argument, let's say 400 nanometers, let's say. So if you shine a 400 nanometer light on this fluorophore, what it does is that it would get excited. The molecules inside the fluorophore would get excited, and it would relax at a different wavelength, and it would give off light at a different wavelength, or in a longer wavelength. So do that in a different color. So this fluorophore, let's say this fluorophore gives off light when it fluoresces at, let's say, 600 nanometers. Just for the sake of the argument, let's say if you shine a light at 400 nanometers on this fluorophore, it would give off light. It would fluoresce at 600 nanometers. 600 nanometers. Now, if you excite, so this is excitation. So this is excitation, and this is emission. So in your microscope, if you set your detector to 600 nanometers, or at the emission wavelength of the fluorophore, if your fluorophore is there in your sample, it would light up like that, right? So if you shine the correct frequency light, and if you collect the return signal at the correct frequency, you should be able to see a signal like that, right? So the other thing is that this fluorophore is actually attached to a complementary sequence, right? It is attached to a complementary sequence of DNA that precisely matches telomeres, right? Telomeres in this case, right? So if telomeres are there, so this sequence goes and binds to telomere regions at the ends, right? At the ends it binds. So if you shine the light at correct frequencies, and if you collect the emission or emitted light at the correct frequency, you should be able to see the fluorescence coming from the ends, because the telomeres are always at the ends of the chromosome. It's not at the middle. It's not at the beginning, but it's always at the end of the chromosome, right? They're always at the end of the chromosome. Right. So if you look, if you could zoom in and look at the end, look at the DNA at the tip of your chromosomes, right? Which is what you have done here. So even if you put your chromosomes under the microscope, you will see exactly the same, right? You will see exactly the same where telomeres would be glowing at the end, right? So what would you see? You might expect to find genes or some DNA sequences involved in regulation. Is that what you would find? No, that's actually not what you would find, right? So you will not find DNA sequences involved in gene regulation or any genes or anything at the end. Instead, what you would actually find is a single repetitive sequence, right? So at the end, what you always find is a single repetitive sequence. So for human beings, it's TT8GGG. TT, so it's a double T, an adenine, followed by a triple G, right? And that's repeated thousands of times, right? It's repeated over and over again, hundreds or even thousands of times, depending on the cell type, right? If it is a skin cell, you have because it divides all the time, right? It divides all the time. So there are a lot more telomeres present on your skin cells than a neuron, right? So your neurons won't divide as much. So telomere regions are not many in your neurons. But if you take a skin cell or any epithelial cell, you have thousands upon thousands of telomere repeats. So the repeating region for human beings is TT8GGG, right? So if you take different species, it differs slightly. If you take a chimpanzee, it's exactly the same as a human being. Right? If you take a gorilla, if I may remember, there's one base change in a gorilla. If you take a cow, it's slightly different. If you take an yeast cell, it's slightly different. If you take an amoeba, it's slightly different. So what I'm trying to say is that regardless of the eukaryotic cell type, the telomere repeats are always there, right? Telomere repeat regions are always there, but depending on the species, there are subtle differences between telomere sequence. So it's wonderful, isn't it? So you have this repeating region, repeating over and over again in the ends of every single chromosome, right? At the end of every single chromosome, you have this repeated region called telomere repeats, right? Now, you might be wondering what on earth are these telomere repeats are doing here? Because it's always present, right? It's always present in your DNA. It's always present in any human being. If you take any human being, the telomere repeat regions are always there, right? If you take any eukaryotic cell, telomere repeat regions are always there at the end of the chromosomes. Now, what it does is an interesting question because it is related to one of the main problems faced in the replication machinery, of eukaryotic organisms. Now, if you take the leading strand, there's absolutely no problem. There's only one primer, right? There's only one primer. It goes from 5' to 3', right? But what about the lagging strand? In the lagging strand, the synthesis is discontinuous, right? The synthesis is discontinuous. So you have to put primers every now and then. So these are RNA primers. There's so this is an RNA primer. This is an RNA primer. That's an RNA primer. Now, so this is the lagging strand, right? Sorry, let me. just to avoid confusion, I'll do that. So this is one replication fork, one replication bubble. So this is the replication bubble, one replication fork moving that way, the other replication fork moving that way. Leading strand, that one, continuously synthesized from 5' to 3'. No problem there. So there's only one prime, right? One primer, one primer. That's all it takes, right? That's all it takes. Lagging strand, however, you need multiple primers. So primer here, primer there, and primers everywhere, right? It needs primers. So these are RNA primers, right? So this one RNA primer in the leading strand. RNA primers. Now, in the leading strand, there's no problem. RNase H removes that, and then DNA polymerase alpha would come in, and it would replace that. Right? It would replace that, and there's no problem. Right? For all the other parts in the lagging strand, same thing happens. RNase H comes in. RNase H comes in and it removes that. It removes that and it removes that. And then it replaces this stretch over here, right? So all the other preceding parts it replaces. But what about the last one? So when it comes to the last one, it lacks a 3' end to attach to, right? So the DNA polymerase alpha lacks a 3' end, because this bit over here is empty, right? This bit over here is empty, so it does not have anything, any 3' region to attach to, right? So what it does is that once it leaves the last primer, right? Once it degrades the last primer by RNase H, it leaves a gap. It leaves a gap like that. In both ends, it leaves a gap. Wherever there's a lagging strand, the last bit is left as a gap. Now, you can see that there's a gap, and there's a loss of information here, right? Now, whenever there are gaps like this, so this is single stranded. So this region, let me use a different color. So you can see that this region becomes single stranded. Right? This little region over here, it becomes single stranded. Now in cellular environments, the cells do not like single stranded areas. Right? So there are nucleases immediately degrading these single stranded areas. Right? Immediately, single stranded areas would be degraded by nucleases. So imagine you have a gene over here, right? Imagine you have a gene, or a part of an important gene. Every time the cell replicates, you lose a little bit of information, right? You lose a little bit of information because the end cannot be replicated towards the until the very end, right? The end bit cannot be replicated towards the until the very end. So for that reason, every time a cell divides, if there's no solution, every time a cell divides, it tends to lose, or at least it sets to lose a little bit of information at the very end, right? And this is, of course, undesirable, right? To say the least, this is completely unacceptable. Because imagine if you have a gene, right? Every time a cell divides, it would lose a little bit of that gene, right? It would lose a little bit of that gene because of this so-called gap, or we call it a primer gap, right? At the 5' end of each strand. However, life is not that stupid, right? The natural world or nature is not that stupid. It has a solution to this problem, right? The nature solution, or the solution of the natural world, is ingenious. It's called a telomere repeat region, right? So the answer of the natural world, or the answer of the natural replication machinery, is called a telomere repeat region. So that's exactly where this TTA-GGG sequence would come in. So instead of having a real gene over here, right? Instead of having a real gene at the end of every chromosome, what it actually has is this TTA-GGG repeat sequence, repeating over and over again, like that. In both ends, you have this telomere repeat region. Now, this is a repeat region, right? It has many repeating units. Each time the cell divides, one repeat region is lost. So at the end of the first division, this part is lost. At the end of the second division, this part is lost. At the end of the third division, that bit is gone. You get the idea, right? Now you get the idea. At the end of each division, a telomere repeat region is lost. Right? The telomere repeat region is lost. So depending on how many telomere repeat regions there are, the cell can divide over a long period of time for a finite number of divisions, or it can undergo a finite number of replications depending on the number of telomere repeat regions that it has, right? So you get the idea about this, and I hope it is really clear. So instead of losing a real part of the gene, it has a pseudo sequence, right? It has an expendable sequence. It has an expendable region that it can actually lose, right? It doesn't matter if the telomere repeat regions are lost, because it does not code for any information. It's just a junk piece of DNA that can actually be lost, right? So these are expendables, right? So these are real expendables in cellular sense. So you get the idea. It's a very wonderful thing, right? It's a very wonderful trick that these cells are actually playing when it comes to cell division. So now I told you that the telomere repeats are being chopped off one at a time. Telomere regions are chopped off one at a time after each cell division. So when a cell begins its life, right? So for an example, one of our skin cells, when we are little, so when we are small, when we are kids, we will have very long or big telomere repeat regions. We will have long telomere repeat regions like that, right? So it's the telomere repeat regions are full, and they're complete. But as we age, right? As we age, when we grow up, they continually or they progressively become shorter and shorter, and even shorter when we get really old, depending on the number of division cycles that our cells undergo. So you can see that the number of cell divisions that cells can undergo is actually finite or limited in a normal cell, right? At least in a normal cell, the number of divisions that it can undergo are actually finite or limited. There are special instances. There are special instances where there are exceptions to this rule. We'll talk about that in a little while. Yeah, so I'm in two minds whether to watch a little animation before we proceed any further, or to discuss about telomerase. Okay, just a moment. Let's watch a little animation before we step into this bit, because this is a little detour from what we have been discussing. So I'll stop here and do a little animation.
Of DNA replication, we have to deal with the end replication problem, which occurs at the end of DNA strand. And this problem is dealt with another process called the telomerase replication in eukaryotes, which we are going to discuss today. In order to understand the telomerase replication, we see we have a DNA molecule unwound into replication bubble by helicase enzyme, MCF. On the upper strand, the primer is added, which provides 3-hydroxyl end for the polymerase enzyme, and it's elongated in the leading strand synthesis. But on the lower strand, having direction 3' to 5' end, multiple primers are added, and the polymerase elongates these primers in the process of lagging strand synthesis, while we get the Okazaki fragments. We have already discussed these processes in detail. Now, what happens next is that the helicase moves to the end of the strands, as shown in the diagram, and unwinds the strand, or we can say separates these strands, like this shown in the diagram. If we draw it out here, we get the DNA strands like this at the end of the replication. Here, the upper strand is continuously being synthesized, as it always provides 3-hydroxyl end for polymerase enzyme to add nucleotides. But on the lower strand, again, multiple primers are added. Do not get confused here. The primers are added one by one on lagging strand when helicase opens the strand. Now, getting to our point. The multiple primers on lagging strand provided these 3 terminus ends shown in the diagram. Then these primers are elongated in lagging strand synthesis, and we get the Okazaki fragments. Now, after that, these primers are removed by the action of RNase H or PAN1 enzyme. Since these are ribonucleotides, they need to be removed from DNA, and we need to incorporate the oxynucleotides in place of these ribonucleotides. So finally, these primers are removed from lagging strand as well as from leading strand. Now let's focus upon lagging strand, because this is why the end replication problem occurs, and telomerase has to work upon that. So after primer removal, we get the DNA molecule like this, having lagging strand with gaps in between segments. These are primer gaps, which need to be filled by deoxynucleotides. So this gap filling work is done by the DNA polymerase. We know all polymerase enzymes add nucleotides on 3 terminus ends, or you can say hydroxyl end. So here we have these 2 gaps where the polymerase can work on these 3 hydroxyl ends and synthesize the strand. This work is done by DNA polymerase alpha. Now we have a problem at the end of lagging strand, where we have 5 terminus ends, and polymerase needs 3 terminus ends to synthesize this part. So this last primer removal on lagging strand always ends up with this problem, which is fixed by telomerase replication. Remember, most of the cells remove the parent strand overhang by PAN1 enzyme, thus creating a DNA molecule. But this way we lose some information, as this is the part of genome. Due to this, at every cycle, DNA molecule shortens by this end replication problem, and we end up losing important information from DNA molecule. And this is the reason of aging also. But some cells in our body have overcome this end replication problem by the presence of telomerase enzyme. The telomerase enzyme has got its own RNA template, plus it has polymerase activity too.
Just a little addition to what the animate is saying. So this talks about there are certain cell types that lack telomere repeat regions. So there are certain chromosomes that actually lack these telomere repeat regions. So telomerase enzyme would come in and add telomere repeat regions to circumvent that problem. So by default, most of the cells, for example, epithelial cells and most other cells in your body would have telomere repeat regions built in when the cells are actually initiated at a very young age. So those are progressively lost, but there are certain types of cells that actually do not have. So either the cells have completely exhausted the telomere repeat regions, or they do not have any telomere repeat regions. So if that's the case, sometimes it can be overcome by this particular enzyme called telomerase. So let's continue to listen.
It can add oxynucleotides to 3 terminal strand. Here, in this case, the telomerase works on parent strand only, while it elongates the 3 terminus end of DNA parent strand. Let's see how this is done by telomerase enzyme. In this diagram, you can see the DNA molecule. It's having a lower parent strand with a sequence of guanine, thymine, thymine, 4-timine guanine, and thymine, thymine, repeated several times. This is called the telomere sequences. And this particular sequence is from holotrichous ciliate. You can see in this chart, these are the different telomere sequences.
Okay, so this is the difference that we have spoken about earlier. So look at this. So for human beings and most of the other mammals, birds and reptiles, is TTA triple G. Right? TTA triple G for humans and most other mammals and birds and reptiles. For trypanosomes, it's TTA GGG again. Ciliates, it's different. And for different type of ciliate, it's yet again different. For yeasts, it's threefold. It can vary. For plants, again, it's different. So it's triple T, A, and a triple G. Right? You don't need to memorize this, but please remember that these are different from different types of eukaryotes when you look at them. Right?
Of different organs at the end of parent strand. Prior to this, we had RNA primer complementary to this telomere sequence, which was removed. And now we do not have any 3-hydroxyl and only deoxynucleotides can be added to complete this end replication. So for this purpose, here comes the telomerase enzyme, which is a reverse transcriptase enzyme carrying the RNA template. The last 2 bases of telomerase enzyme pair up with the last 2 bases of DNA strand, and then drives the DNA synthesis. And then again, it pairs up with the terminal 2 bases and drives further synthesis, thereby elongating the parent strand further. This highlighted DNA part, as shown in the diagram, is the newly synthesized DNA by telomerase enzyme. And to its right, we have the actual parent DNA. That is the part of genome. But again, we cannot synthesize the upper strand because it's devoid of 3-hydroxyl end. But here the primase comes in and lays down the primer at the end, thus provides 3-hydroxyl end. Now polymerase alpha, or you can say DNA polymerase alpha, comes in and starts adding oxynucleotides to the 3-terminus end of primer. And by this, the upper strand is synthesized. Now again, we are back to the same situation. The RNA site works on the primer and removes it, since it's the ribonucleotide sequence. So again, we have same daughter strand missing the last few nucleotides. But here now we can remove the parent strand overhang flap because this is junk DNA synthesized by telomerase enzyme. We only need this part of DNA on the right. So N1 comes in and cuts down the overhang flap at the end of telomere. So with this, we do not cut the DNA of organism. Rather, we cut the newly synthesized DNA called the telomere. Again, I repeat, in the end, we can say the telomerase replication saves the tail or end of it. In the end, when primer is removed, there will always be overhang flap. So we need to cut that overhang flap at the end of replication. But this is part of genome, which cannot be removed as such. So what this telomerase enzyme does is that it will.
So you get the idea, right? So that's how the telomerase would act upon the final few bases, and then it would act. It would add telomere repeat regions repeatedly if it is necessary, right? Certain cell types would have telomerases, right? Telomerases would be present in certain cell types, but not all cell types, right? So that means, for an example, neuron cells, they do not have a very good telomerase, so it can only undergo cell division for a limited number of cycles, and then the cell division or the replication would stop, right? So you can see how the end replication problem is overcome by the action of telomerase, right? So it has an RNA template within itself. So it's a ribo nucleoprotein. So that means it has a little RNA part. So ribo is the little RNA part. It has a little RNA template. RNA template, which is this one over here. So it's a protein, but it has a little RNA template like that, right? So it can basically pair up with the last telomere of the chromosome, and then it can elongate the telomere. So you can add telomere repeat regions to the telomere. Yeah, so I hope it is reasonably clear. So so the same process that was described in the animation is described over here using different bases, of course. It's a different telomere repeat region that's shown here. But the idea is the same, right? So it pairs up with the last two bases like that, pairs up with that, and then it starts to add a repeat region repeatedly like that, right? So like that, you can add the repeat region towards the end. Now, speaking of the importance of this, this telomere, so where should I start? Yeah, so I'll speak about aging, right? So so I told you that certain cell types do not have a good telomerase, and it cannot repeatedly add telomere repeat regions indefinitely, right? Some cells have a limited telomerase activity, so it can add telomerase for a certain amount of time, and the telomerase activity would would be lost. So in that case, you can have a finite number of telomere repeat regions, but not an infinite number. So what it means is that that particular cell type can undergo cell divisions for a finite number of times, and then its replication or its growth would stop. Now, what this means is that once you reach a certain age, your telomere repeat regions will be exhausted, right? Your telomere repeat regions will be exhausted, and your cell, that particular cell type, will remain undivided, right? So they will stop dividing, they will stop replicating, they will start to age, right? So the cells would start to age without replication. So no new cells would be replaced if cells are damaged due to metabolic processes and things like that. So it would basically come out of the cell cycle, right? So when the telomeres are exhausted, when the telomerase is not acting, when there are no free telomere regions are available, the cell would simply come out of the cell cycle, right? It would stop dividing. It will still function. Right? It will still function. It will carry out its function, unless the cell is fully damaged and it would die. It would still function, but it would function at a reduced effectiveness because the cell would start to age as the time goes on, right? So as a result of that, we now know that telomere shortening. So I have shown you how telomeres would shorten at the ends of chromosomes like that, right? So we have long telomeres when we start our lives. They would progressively become shorter and shorter and shorter, and eventually they would completely disappear. When we age, when we come into a mature age, they would completely they would be completely gone when we come into a mature age, and the cells would be locked in the cell cycle without further dividing. So when this happens, the cell would start to age, right? When the cells are aging, what it means is that the organism would will also age, right? So the owner of these cells are, if the owner of these cells are us, what it means is that we would also start to age, right? Because our cells are now aging. So now we know that telomere shortening is one of the major drivers of aging, right? That's one reason why we age. There are other reasons that we age. There are toxic metabolites that are accumulating inside cells. There's reactive oxygen species damage that our cells are taking, when we reach a mature age. But one of the major reasons that we age is actually telomere shortening, right? So telomere shortening would lock up most of our cells in a mature state, right? In a mature state that are incapable of further dividing and replicating themselves. Therefore, when the cells are locked in this cycle, locked from the cell cycle, we start to age, right? Start to age. First, our cells age, and then ourselves will start to age, right? So that's one of the fundamental things about life, right? It's directly linked to genetics and biochemistry, right? So people think, oh, we age and it's inevitability. It's inevitable. Yes, it is inevitable. As a process of life, aging is inevitable. But you should keep in mind that it's a genetic process. First of all, it's a genetic process, and second of all, it's a biochemical process. And most of the things are poorly understood at the moment, but we are getting a complete picture, even now, right? When the science is advancing, when new knowledge is becoming available, now we now know that telomere shortening and other mechanisms are directly culpable in the process of, or directly responsible in the process of aging. Right. So cells can undergo cell division to continue to have their telomeres shortened, because most somatic cells do not make a telomerase, right? So what it means is that they can undergo a finite number of divisions. They can undergo a finite number of divisions before all division stops because of a lack of a telomerase, right? So they have a finite number of telomere repeat regions. When we start off, they would shorten and shorten and shorten progressively as we age, as we reach a mature age. So once it reaches the end of the telomere repeat region, it would stop dividing completely. So this essentially means that the telomere shortening is associated with aging for this particular reason. In 2010, just 12 years ago, scientists found that telomerase can reverse some age-related conditions in mice. Right? So if we think death and aging is inevitability, well, you have to think again, right? Because some of the aging related conditions or aging related phenomena can be reversed with the help of telomerase. We do not know how to do this exactly at this point, but it does not mean in the future we will not be able to do that, because we are continually learning about this, and some of the age related effects or aging effects can be reversed, at least in mice, right? So that means we are now gaining a much more thorough understanding about how telomerases are engaged in the process of aging, right? So it's not really inevitability. I mean, of course, for the cyclic nature of life, birth and death are essential. I completely agree with that. But just for the sake of the argument and just for the proof of principle, so if you think aging cannot be reversed, that's a wrong perception, right? So aging and maybe even death, it could be very substantially prolonged. So age of a person may be very substantially prolonged in the future. I'm against that whole perception, because it goes against the continuity of life. So if an animal or a person is born, that has to die, right? So that cyclic nature of living things would continue. I'm completely against that principle, but as a proof of concept, I'm saying that it can be done, right? At least in the next 30 to 40 years, there would be signs of that happening. So already there are signs of that happening. There's much more progress from here. In 2022. So even in last year, 2021, there were major breakthroughs in terms of aging and the action of reactive oxygen species, for an example. So we call them ROS. So the generation of reactive oxygen species inside the cells as part of cellular metabolism. So for an example, in mitochondria, we accumulate a lot of ROS as a result of cellular metabolism. So in 2021, there were findings to suggest that ROS accumulation can be reversed, and therefore aging-related phenomena can also be reversed to a certain extent by this process. So likewise, telomere shortening can be reversed by the introduction of telomerase into certain cell types, and of course, aging can be reversed, or at least age of an organism could be significantly prolonged, at least in principle. Right, so that's the role of telomere shortening and telomerase and telomeres in the process of aging. But there's another nasty thing about telomerases. So this is the good thing about telomeres and telomere shortening and telomerases. It would allow eukaryotic cells to go on dividing and to refresh themselves. But there's a darker side to this. Telomerases, right? So in particular, telomerases. Telomerase. What if your telomerase becomes uncontrollable? Imagine a scenario where your telomerase is uncontrollable. You have a version of your telomerase. It's continually and very aggressively adding telomeres to your cells. What could happen? Can someone very quickly tell me? I do not want to spend a lot of time, but you can use your imagination and very quickly tell me. If your telomerases are very aggressively adding telomeres to your cells, what might happen? Any ideas? Can someone tell me? Can someone tell me a type of cells that are very, very good at dividing, or maybe I should say a type of cells that are very aggressive in dividing? You know where I'm going now. A type of cells that are very aggressive in growing and dividing themselves, or a type of cells that are very aggressive in replicating. What could be that? Quick answer would say that again a little bit louder.
Cancer.
Yes, absolutely. Tumors and cancers, right? Tumors and cancers are very good at dividing themselves, right? They are very good and they are very aggressive, right? They very aggressively divide and replicate themselves. And make no mistake, they have a very good telomerase. Cancer cells. Or sometimes you know, a mild or abnormally dividing cells. We refer to them as tumors. Cancer cells or tumor cells, they have a very, very good telomerase, right? It's very aggressive, right? Very, very aggressive to the point where it's abnormal, right? All it wants to do is to grow and divide, right? It doesn't even wait until you know, oxidative phosphorylation. It doesn't even wait until that. It only tries to get energy using glycolysis, right? So it does glycolysis because it wants to, all it wants to do is to divide, right? So cancer cells, it has a very, very aggressive and a very strong telomerase, right? So that's why it's very good at dividing. So it continuously adds telomere repeat regions at the end of the chromosome, so that the cancer cells can, you know, it can basically grow and replicate themselves like crazy, right? Until before we know, you know, we get a cancer. So you have a tumor, you have tumor cells growing and growing and growing, and once it reaches to a certain threshold, before you know it, you know, there's a cancer. So there we go, right? Telomerase, it can help our cells grow and it can help our cells replicate and divide in normal circumstances, but it can go, it can turn weird, and it can also give rise to tumors and then ultimately cancers. And one big part of cancer biology is actually telomerase, because without the telomerase, the cancer cells are unable to grow. So up until now, the attempts to repress cancer telomerases have not been very successful. There are anti-cancer agents that would target telomerase enzymes, but you know, that they haven't been very, very successful, because the telomerase that the cancer cells have are very, very strong, and extremely strong, very efficient telomerase, right? So I think that's where I would like to stop, because no, a few more slides. Just I'll be quick with this. Now, this particular slide is very, very important for you. So here it's a contrasting of DNA replication between eukaryotes and prokaryotes. So pay good attention to this one. So it's an important slide for you to take a look at. Now, the last thing that I would like to touch upon is this one, derivations of DNA replication. I will not spend too long on this. I will invite you to do a little bit of self-study on this aspect as well, but I will give you a little bit of information about this. So the derivation that I would like to draw your attention to is something called mitochondrial DNA replication. So in mammalian mitochondria, they contain multiple copies of circular double-stranded DNA genome and a dedicated replication enzyme, first of all. So that one is DNA polymerase gamma that we have discussed very early on. So it has a separate dedicated DNA replication machinery for its replication and maintenance, right? So mitochondrial DNA is double-stranded molecule of 16.6 kilobases. So the two strands of mitochondrial DNA differ in their base composition slightly. They differ, with one being rich in guanines, and that one, for that reason, it would make it a heavier strand. So we refer to that as the H strand, and the other strand, the normal strand, we refer to that as the L strand or the light strand. Actually, if you denature the two copies, and if you do an ultracentrifugation called cesium chloride ultracentrifugation, density gradient ultracentrifugation, you can actually separate these two H and L chains. So this is a centrifugation done at a tremendous speed using a cesium chloride gradient. So when you do that, you can do very fine separations in molecules. So once you do that sort of a separation, you can actually separate the heavy chain from the light chain. So the replication of mitochondrial DNA progresses in the form of a so-called D loop, which looks a little bit like this. So for this reason, we call it a D loop, like that. So I told you that I invite you to do a little self-study. So this is, I do not expect you to memorize this material or whatever. In fact, I do not, expect you to memorize anything, right? I would expect you to have an understanding about the concepts that we discuss. Don't try to memorize things, right? Memory is inferior to understanding, right? Understanding is the superior thing, right? Understanding is a superior concept, and memory is the inferior concept, right? So if you try to memorize things, it's not desirable, right? It's not desirable. The desirable thing is understanding. Try to understand these concepts rather than memorizing it, because memory can escape your mind very easily. Understanding will not. Understanding will stick around, right? So if you understand a certain concept, for an example, eukaryotic replication, if you get the mechanistic understanding of, if you get a proper mechanistic understanding of eukaryotic replication, for an example, it will stick around, right? It will stick in your mind for a very long period of time. And if you, as opposed to that, if you try to memorize things, you're a loser, right? I mean, you lose out on a lot because you can escape your memory very easily. So try to gain a good understanding about eukaryotic replication in particular. And this one is not so important. It's just a derivation of replication that's taking place in mitochondria. So what happens mechanistically is given over here. So do take a read in that one, and don't try to memorize anything. Just try to get a little understanding about how this takes place. It's called D-loop replication because of this particular shape that it's taking at the start of the replication process. So likewise, you have the heavy chain and the light chain that I described earlier on. So there's a description of this particular process over here. So that leaves us at the beginning of the DNA repair mechanisms, which I will do the next week. So that means we've come to the end of the lecture. So before I ask you about any difficult areas or any questions that you would like to ask me, I would like to also ask whether you have any problems with the Google Classroom. If you have anything, so last week someone told me, there was a problem. I rectified that immediately. So I'm sure that I'm pretty certain that it's resolved at this point in time. If not, you better tell me as soon as possible. Does anyone have any problem with the Google Classroom logging into that and using that, operating that, or anything of that sort? You can immediately tell me. No, I think there's no issue. It seems everybody's quiet, so I would assume that that is not the case in terms of problems. So the second thing that I would like to ask you, do you have anything to ask me about today's material that we have covered in terms of eukaryotic DNA replication? Any trouble spots or anything, any concerns, or maybe any curiosities, anything that you would like to know additionally, anything additional that you would like to know? So we talked a little bit about aging, and we talked a little bit about the role of telomerases in cancer, and how cancers become very aggressive. One of the reasons is actually telomerase, right? One of the reasons why cancer cells and tumor cells are very aggressive is that it has a very efficient telomerase, much more efficient than our normal somatic cells. That's one of the reasons why it's very aggressive. So likewise, if you have any questions, you can ask me. No, nothing it seems. So that means we've come to the end of the lecture. So thanks a lot. I'll see you next week again at the same time at 8 o'clock on Tuesday for a brand new lecture. So thanks a lot, and see you next week.